All right, everyone, we're going to go ahead and get started. Turn that down just a little bit. I just want to say thank you, everybody, for being here for our research seminar today. If you are new to this event, these are hybrid events, so we also have folks online. So welcome to everybody online as well. My name is Cinnamon Moffett. I'm the research program manager here at Oregon State University's Hatfield Marine Science Center. I, along with somebody I'll introduce here in just a second, will be the host for this event today. Just for folks online, I wanted to let you know that we have a volunteer in the room. Her name is Roxanne. Roxanne will be able to answer any questions you might have if you're having any technical issues. She will also be able to read out any of the questions you have for today's speaker so that folks in the room can hear and follow along with the questions that you all are giving us, and our speaker will be able to answer those. For folks in the room, we ask for you to raise your hand and I'll bring you a mic, and you can ask your questions that way as well. But again, thank you so much for being here and being a part of this event. I have one quick announcement and then I will introduce my co-host. So next week's speaker, April 25th at our seminar, will be Maggie Somers, who is NOAA's West Coast Sustainable Fisheries Division. We'll be talking about assessing and improving equity in federal fisheries management. So that is a really interesting topic. So I'm really excited to have Maggie come and join us. So I'm hoping you can come back and join us next week. If you are interested in any of our HMSC events, you can go to the HMSC homepage, scroll to the bottom. There is a calendar there, which you can get for any of our events. You can get all the information you need. But I'm going to get this started. I'm going to hand this off to Keith. Keith is a postdoc here who invited our speaker. So I'm just going to hand the mic for Keith.
Thank you, Cinnamon. So I am absolutely delighted to welcome today's seminar speaker and my previous postdoctoral advisor, Dr. Roxanne Beltran at the University of California, Santa Cruz's Department of Ecology and Evolutionary Biology. Roxanne got her bachelor's at UCSC and then a master's and her PhD from the University of Alaska. Roxanne then postdoced at the University of California, Santa Cruz under the supervision of Dr. Dan Costa. And her research group is interested in the adaptations of organisms in the marine environment, especially the movement, foraging ecology, and energetics of pinnipeds, cetaceans, and seabirds. They seek to understand when, why, and how animals do what they do in the context of their environments, and have developed a set of innovative tools to explore the drivers and consequences of life history events and to tackle big questions in ecology. And with that, Roxanne, the floor is yours.
Thank you so much, Keith, for that wonderful introduction. Can you give me a thumbs up if you can hear me? I'm not talking into the void. Can you hear me?
She can't see the thumbs, so I'll say thumbs up.
Okay, perfect. Thanks. All right, I'm really excited to share with you all some of the research that my lab has been working on, thinking about how open ocean ecology shapes life history evolution in really long-lived mammals. Today, I'm going to be talking a lot about the elephant seal system that I work in, which is what you see here at Año Nuevo Reserve in California. So to start, I want to think really broadly about the ecological impacts of living animals. When we think of animals, we know they're often migrating, they're foraging, they're molting, they're reproducing, and they're socializing. And we can think of their ecological impacts in really simple ways, right? Like how they cycle nutrients, how they deplete prey resources, or how they offload contaminants, the role that those behaviors play in things like gene flow and in pathogen transmission. But obviously, for those animals to have those ecological impact, they have to be alive. And my group thinks a lot about how it is that animals stay alive. So I want to do a little pop quiz. I'm really sad I'm not there in person to do this with you guys in real life, but I want to have you guys think and reflect on how animals die, because I think it's something we overlook quite a bit in research. And we have some sort of either like biased or some sort of, I don't know, pre-existing ideas of how animals die. Like maybe if you've seen my octopus teacher and I show you this picture of an octopus and ask you how, you know, these animals typically die, whether it's predation or starvation or old age, you might think of old age or senescence as a reason that a lot of, you know, octopuses die, right? They breed and then they die. What about impalas? If you've seen David Attenborough on Netflix, if I ask you the same question, you might not think about old age as a predominant driver of mortality. You might tend to think of something like predation. And if I show you this picture of a polar bear and ask you the same question, you might think of starvation, right? We've all seen the picture of the polar bear on the ice floe because of climate change, et cetera, et cetera. And some of those assumptions or those sort of predetermined beliefs are totally legitimate, and sometimes they're not. But the real answer is we're lacking a lot of good information about how wild animals die. There was a really nice review by Hill and colleagues in 2019 for terrestrial mammals and birds that showed the causes of mortality in a whole slew of, over 100 studies, and showed basically that predation plays a huge role in how wild animals die. Human harvest also plays a pretty substantial role, at least in the studies that have been published about it. And starvation supposedly plays a pretty limited role. But unfortunately for marine vertebrates, we know a lot less about how animals die in the wild, and therefore the sort of selection pressures that are acting on them, whether that's starvation or predation or human harvest. One of the sort of related questions to that is not just how they die, but also how long they live, right? So if we think about some sort of wild marine mammal like an elephant seal, we know on average they tend to live many fewer years than their potential maximum lifespan. So elephant seals on average live to be about 1.4 years old. They can live to be 23 years old, but that's extremely rare. And the same thing is true for lots of species, like this little brown bat that tends to live only 2.2 years on average, but can live to be 34 years old. And this is really different, right, than the sort of domesticated animals and like humans that tend to live on average a really long proportion, a really long lifespan relative to what they could possibly live. And so comparing these makes it really obvious, right, that wild animals face a lot of really strong selection pressures. So if that's the case, then what can animals do, whether it's behavioral strategies or some sort of like physiological adaptation, um, that allows them to stay alive for longer and therefore contribute genes to the next generation? If we look at wild animals generally, we see a huge amount of variation across individuals. I feel like the among individual variation is becoming much more obvious, and people are accepting it as much more ubiquitous than we did before. So things like anatomy, like body size, are really different across individual animals, even for the same age. We know that physiology can be super variable, even within a given species. We know that behavior and ecology and evolution is super variable among lots of individual animals. So one of the focuses of my lab group is thinking about this variation and trying to tie it to demography, trying to figure out if there's certain strategies or certain timing or certain bits of performance that really relate to survival in wild animals. And one of the tools we use for that is biologgers, or instruments attached to animals. So there are a million biologging studies going on all over the world by tons of different research groups. My group in particular pairs that biologging with demography, but biologging in general is an incredible tool because when animals die, they tend to go offline. So there will be some sort of Argos-linked satellite transmissions that all of a sudden disappear when an animal is no longer alive. I just want to put a little shameless plug in here that I gave a talk at the biologging conference that was in Tokyo earlier this month, I guess it was last month, about the sort of potential benefits of biologgers for producing really impactful ecological insights. So this sort of figure on the left shows different animals across walkers, swimmers, and flyers that can be instrumented. The insets show different scales of biological organization, from individuals to populations to communities to ecosystems. And all the colored dots here are different parameters that can actually be measured by biologgers to give us insights about how animals operate, from the individual to population level, et cetera. And then the white boxes show ecological theories that can be tested using biologgers, ranging from things like risk-reward trade-offs and how animals navigate them, to things like landscapes of fear. And you'll see some of these ecological theories sort of being tested in the case studies that I show you today. If you are interested in that paper, it's currently at EcoEvo archive as a preprint. It's in review at Trends in Ecology and Evolution, and I'm happy to send you that paper if you're interested. Just let me know. But I think that's a really sort of nice framing for the type of work that my research group does. And then the system that we work is in California, as I mentioned before. So these are northern elephant seals. They come to the beaches twice per year. They have offspring that are on the beach, and then we can very carefully capture the pups, like that one in the front that you see there. We can weigh the pups to get a metric of both maternal foraging success, because the moms are nursing those pups and raising them to different sizes, and also an indication of the ocean health. We can put flipper tags in those offspring so that we can uniquely identify them for the rest of their lives. And then we can watch them every single day for both their breeding and molting seasons when they come on land, throughout their entire lifespans. And that's how we know that elephant seals can be 23 years old, but that most of the time they only live to be, 1 or 2 years old. I want to show you a little bit what this looks like. So this is a video we did with the BBC. You can see that seal has a mark on her left side. That's just a hair dye mark with her individual flipper tag number. You can see how easy it is to see the pups associated with the mom. So we can also get things like pedigrees to be able to know mother-offspring relationships. And we can literally walk around the seals. We can do those observations. We can see which ones are alive, which ones reproduce, and what they're doing. So we can get a really good sense for their demography and also for their behavior. And so this is a little bit what that looks like. There again is a marked seal, B95. She was born in 2015. She was sighted 5 times during her birth year, and she's now 9 years old. But at the time I took the screenshot, she was 7. And we've seen her, over 100 times. So we can get all sorts of really useful information about her, including, her reproductive success, her life history timing, and her survival. And this program has been going on for 6 decades. There are now 360,000 sightings of 50,000 seals. So really rich information about all sorts of things from demography to behavior linked to demography that we get using those biologging instruments. So this is an adult female carrying a satellite tag that can give us information about where she's going, how deep she's diving, and also about the ocean conditions that are around her. And it's not just that we can get behavior and demography, we can also get feeding success. So when the animals leave the beach, they're super skinny. When they come back from their foraging trip, they're nice and fat. We can weigh them both times and calculate the foraging success during the trip as mass gain in kilograms per day that they were out there in the North Pacific Ocean feeding during those foraging trips. One of the things that we think a lot about in my group is the impacts that our research is having on the animals that we studied. And I just want to give a shout out to Allison Payne, my PhD student, who did a really beautiful paper that's in review right now on the impacts of biologgers on walkers, swimmers, and flyers, and how they can be strategically reduced. So she reviewed the literature to look at whether studies reported instrument impacts or reported no instrument impacts or didn't report any instrument impacts at all, and then kind of partition that among taxa to make some recommendations for best practices for instrumenting animals. And that's also available as a preprint. So let me know if you're interested, and I'm happy to send that link. But the point is, we try to use best practices when we're instrumenting the seals. So we minimize the tag size. We try to use shapes of tags that will reduce the drag that the seals are experiencing. And we don't think that the instruments that we attach to the animals are influencing their behavior or their demography. I also want to make a quick plug for the field assistants, the undergraduate students, and also the graduate student field leaders who I have the great pleasure of working with. Our lab worked on a paper about how long-term studies like ours can provide this sort of training structure for inclusive education and professional development, ranging from, fieldwork and safe and trustworthy spaces to doing things like presenting at conferences for the first time with existing data, to feeling comfortable in the field because of the infrastructure that's available for the students, to undertaking publishable independent research. So we've been really proud to sort of try to figure out how to best leverage the infrastructure our program has to promote those inclusive training experiences. And again, if anyone has any questions about that, I'm happy to chat about that. Okay, so let's get into the hardcore research. So today I want to tell you 3 stories about sort of case studies that we've done. The first is thinking about the fitness consequences of life history timing, what happens when animals fall behind. The second is about the fitness consequences of behavioral strategies. How is it that animals avoid mortality, and what are the predominant causes of mortality? And then the third is about the fitness consequences of ocean conditions. So how much variability is there really in the North Pacific Ocean where these seals are feeding, and what effect does that have on their ability to produce offspring? So I'll start with this first sort of case study. I really love thinking about the annual cycle. I think we tend to see animals, doing these daily and seasonal activities like migrating and sleeping and foraging and reproducing and molting. And a lot of the approaches that we typically use for research are cross-sectional, where let's say we put up a camera trap and we're, seeing different animals, or we put out a passive acoustic monitor and we're hearing lots of different animals. And that's super helpful for describing basic things like when a particular season is for a species, but it's harder to understand the individual based ecology and evolution. It matters a lot for animals whether timing is synchronous or whether it's staggered. So in this particular example, you can see 3 individuals, A, B, and C, that have either synchronous timing or staggered timing. We know that the ecological consequences are pretty dramatic. So when animals have staggered timing, like let's say animal A leaves a place before animal B even gets there, those animals don't transmit pathogens to each other usually, they don't compete for resources, they don't deplete the prey, etc. And when timing is really staggered, you have to be careful to use longitudinal sampling, right? Because if you pull information from multiple individuals and stick it together like a Frankenstein approach, then you're not getting information at the individual level. You're getting a broad understanding of when a season is. On the other hand, synchronous timing does things like facilitates information transfer between animals. It can facilitate gene flow and communal feeding and, among other sort of important ecological patterns. And it also means that cross-sectional sampling might be fine if we want to, describe roughly the individual timing of a system. And so that sort of framework got me thinking a lot about the molt in elephant seals. So if you have a cat or a dog at home, you probably have seen them shedding. They replace their fur and skin every year, and that is the process of molt. Seals and sea lions do the same thing. And that molt has to fit into the annual cycle. So this is the annual cycle of an elephant seal. You can see where we are in April right now. The seals are just starting to molt, and then they're going to finish their molt pretty soon, depart for a long trip to sea. That's about 7 months. Then they arrive for the breeding season, they give birth, they wean their pups, and then they depart for a short trip to sea before the molt. And when they haul out on the beach at Año Nuevo, we can really watch them molt because they not only shed their fur like cats and dogs, but they also replace their skin. It peels off in these kind of crazy stinky sheets. So we can really clearly see, when they've started that life history event. So the picture on the bottom is a 5% molted seal. You can see that like old brown fur kind of molting off. And then you can see the silver fur coming underneath when the seal's 50% molted, etc. And so we can walk around the beach, read the flipper tags and marks of individual seals, mark down the proportion of their body that has molted, and then we can take those data from an individual seal over time and build this molt progression curve where we can actually mathematically extract the start and end of the molt and also the arrival and departure date based on when the animals hit the beach. And if we take those 4 data points, the arrival, the molt start, and departure for lots of animals, I think we have over 1,000 animals in this paper. This is a subset. You can see that it's pretty staggered, right? So some individual seals actually depart the beach before other seals ever arrive. And that was really surprising to us. We had no idea how staggered this process was. And we also were really surprised that the molt itself only lasts 7 days. So it's a really, really quick process, but it happens in this really big haul out period. We don't in general know a lot about the molt. So we actually just published this paper yesterday. It just came out in Proceedings B, showing the individual variation in that annual cycle timing, talking about how present the seals are in like a synchronous way, how asynchronous the actual events are, and then how they actually rush to catch up if they start running behind in their annual cycle. But it wasn't just the sort of means that we were interested in, it was also the standard deviations. And it was also the gaps between the pieces of the annual cycle. So in a sort of related piece of research, we wanted to ask what is the entire annual cycle of an elephant seal? This may sound like a really simple question, but in many marine mammals, like we just don't know what these annual cycles look like, especially not in a sort of longitudinal way. And so some of the pieces of this puzzle had been put together, but we were missing one, which was how long after arriving from their long migration do they actually give birth to pups. This is for adult females. And what we found is that it's actually only 5 days. So these animals are undertaking these incredibly long migrations. They're 7-month migrations, and they're arriving to the beach in California with just 5 days to spare before they give birth to that pup, which shows you the incredible sort of navigation skills that they have. It looks like they have really particular abilities to navigate back and to strategically return to that beach too, because if you look at the population level standard deviations for the different components of the annual cycle, we show in that paper that the breeding season arrival is extremely synchronous and the molt is a lot less synchronous. So it seems to be that the molt is a much more flexible time that they sort of use as a buffer to, like I said, catch up when they fall behind. But then by the time they depart for that long trip and come back for the breeding season where they give birth and wean their pups, they're a lot more synchronous. And we think that facilitates their reproductive success and the sort of polygynous breeding system that they have where, one single adult male who's an alpha will hold a harem of, a couple hundred seals at most. So the question then is, if they're able to get back so synchronously, then what navigation mechanisms are they using to make those decisions? So we were really interested in figuring out what the return trip looks like. So here's them going out to sea. You can see them foraging a little bit, and then they all turn around and come back. That was the short trip. They do the same thing for the long migration, and then they come back. And I had a senior thesis student, Alex UN, who took a look at this question, and he basically picked out turnaround points, like when the seals started coming back by plotting for each of the seals distance from the breeding beach as a function of time, where 0 on the bottom right of the plot is the date of birth. So going back from that, and basically found like the last point at which the seals were moving away from the colony instead of towards it, and counted that as the turnaround point. And then he mapped those turnaround points in space and found that they were super randomly distributed. So it's not like all the seals turn around, when they're at 160 degrees west of longitude. And likewise, the turnaround points were really randomly distributed in time. So in this plot, the y-axis is, again, time, days before parturition, where 0 is that birth date, and those green dots are arrival at the breeding colony. Each x-axis tick is a seal. And so you can see how random the turnaround points are. So if they're not turning around at a certain place and they're not turning around at a certain time, then what exactly is it that they're using as a mechanism to make it back to the breeding beach? Well, we found that seals that foraged farther began their turnaround earlier. So this is a plot of turnaround distance from the colony on the x-axis and turnaround date on the y-axis. And you can see that negative relationship. So it seems like the seals basically know not only their distance to the breeding beach, but also the time it will take them to get back. And they strategically turn around at the right moment, which allows them to make that breeding synchrony happen. I was pretty excited about the senior thesis project because it sort of showed not only that they have what we call a map sense, which is like animals' ability to have an internal GPS that they can use to know their distance and time, and not just that, but that relative to where they need to go. But it also tells us a little bit more about their sort of navigational abilities, which is one of, I would say, the most elusive research questions in our system, how it is that these long distance migrators are able to find their way home in a featureless ocean. So we were pretty excited about figuring that out and learning about how it facilitates synchrony in some parts of the annual cycle. Okay, so that was vignette number 1. So we talked about life history timing. Now we're going to talk about fitness consequences of behavioral strategies. And I'm super proud to share this particular case study because Keith was my first ever lab member at UC Santa Cruz when I started my lab during the pandemic in 2020. We unfortunately didn't get to interact very much in real life. We finally had dinner a couple years after he left my lab, which was amazing. But we were able to produce this really special paper together that made it onto the cover of Ecology Letters, which is about how behavioral strategies are important for both foraging success and lifetime fitness in these northern elephant seals. So I talked a little bit about individual variation in behavior and stuff like that. And I want to tell you more about individual variation in demography, because that's, again, something that's really hard to study unless you have this like known age, known demography, individual based system that we have with the elephant seals. I'll give you an example of what an encounter history looks like. So let's take animal J493. She was seen every year of life from when she was age 3 to when she was age 20. Well, she wasn't seen. We have data from each one of those years. So if she was not seen during that year, if she was missing, there's a 0 here. If she skipped breeding, there's a 1. If she was seen and produced a pup, that's a P. This is an encounter history that can then be used to calculate lifetime fitness metrics. So how long did she live? 20 years. And how many pups did she produce? Or what was her lifetime reproductive success, or LRS, which here was 5 years old. And we can do that for this seal and another seal and another seal to see that there's a lot of variation in their reproductive strategies. So some of them live a really long time, but produce a small number of pups. And some produce that same number of pups, but live a much shorter period of time. Here's what that looks like in sort of biplot space, where the x-axis is lifespan in years, the y-axis is lifetime reproductive success, and that white line is a one-to-one line that basically represents seals that reproduce every possible year after sexual maturity. So there are those 3 seals, same lifetime reproductive success, really variable lifespans. And here are all the seals in the long-term study. So again, this complete, gunshot plot of all the seals and all their individual variation and how successful they are. And so Keith and I were really curious about whether we could use behavior to explain some of the residuals in this fitness plot. So we looked at a couple metrics. We looked at foraging depth of each seal. So here each dot is an individual seal. We looked at how much body mass they gained during the foraging trip. We looked at where they foraged in terms of distance from the coast, their departure date, and also the proportion of fish in their diet. And what we found is that there are sort of these suites of behavioral strategies where seals that dive deeper also forage farther from the coast. They have more fish in their diet and they gain more mass. So there's these sort of behavioral strategies that can lead to mass gain. So then the question becomes, what's the relationship between mass gain and survival and reproductive success? So here's a plot of the annual probability of survival on the y-axis against mass gain on the x-axis. Here we've actually plotted separately different ages, so you can see that age 15 is much lower than the rest, implying a bit of senescence in this species, which is pretty cool. And overall, you can see that seals that gain more mass have higher annual survival rates, although it's pretty marginal increase. 100 kilograms of mass gain just increases the seal's probability of survival by like 15%. But the annual probability of reproduction has a much more dramatic and much more threshold shape relationship, where a difference in about 20 kilos is the difference between these seals producing a pup and not producing a pup. And I'll just point out, we have some repeated measures of seals who were instrumented and tracked with flipper tag mark recapture in multiple years. And so, for example, 0651 is the sort of diamond shape, and you can see when she gained less mass, she did not produce a pup. When she gained more mass, she did produce a pup. And so we call this sort of a physiological tipping point, where a small amount of foraging success can lead to a really dramatic change in the annual probability of reproduction, and a little bit less so with survival. So putting this all together mathematically into a lifetime context, here again on the x-axis is mass gain, on the y-axis is lifetime fitness. Here's reproductive lifespan, so lifespan after the age of sexual maturity around age 3. And here's what lifetime reproductive success looks like. So if seals gain, let's say, more than 280 kilos in a foraging trip, their reproductive lifespan and lifetime reproductive success actually converge because they're no different. If the seal is alive, they'll produce a pup. Lower than about 200 kilograms of mass gain, those lines diverge. So even if seals are alive and they have pretty long lifespans, their lifetime reproductive success is still not high because they're basically kind of selfish moms. If they're alive, they're just not reproducing a pup. And then there's that sort of intermediate point again where that threshold lives of little bit of change in mass gain will have a big impact on lifetime fitness. So to summarize all of that, we found that there are behavioral strategies. They increase reproduction and survival through mass gain. We know that these particular animals do a little bit of, we call it intermittent breeding. That's not the best term. It's more like skipping reproduction sometimes. And we think that that has to do with how much mass they're gaining. So it's possible that that's sort of a strategy in these guys to skip reproduction in years where they're not gaining as much foraging success. And that probably has a lot to do with their behavioral strategies as well. So that sort of indirectly got into, survival and reproductive success. But we wanted to dig a little bit deeper and see how sleeping at sea is playing a role. So we know when the seals are during their foraging migrations, they're either foraging or they're resting. Those are the two options. When they're foraging, obviously, it's a really high reward activity. They have a lot of energy intake from the fish and a little bit of squid that they're eating. Or they're resting, in which case they're experiencing really high risk. They sleep with both hemispheres of their brain, just like us. So they can't actually detect any of the predators that are coming to try to eat them, like sharks and killer whales. So resting is a really risky strategy in these guys. And so we wanted to see, what is making them forage and what is making them rest. One of the hypotheses that we had is that time of day would play a really important role. Sharks and killer whales are, for the most part, pretty visual predators. And so they use, things like silhouetting with daylight to be able to detect and attack their prey. And so theoretically, the nighttime is more safe for elephant seals and the daytime is more risky for elephant seals. But there's an added complexity in the three-dimensional environment that sunlight also changes a lot with depth, right? Where deeper depths are more dark, so theoretically more safe, whereas shallower places are more risky. And so theoretically, the predators should be taking elephant seals a lot more in the daytime than in the nighttime. And so we were testing state dependence with this idea, trying to figure out if body condition played a major role in how the seals are sort of navigating this landscape of risk, how they're using time of day and how they're using depth. And so I'm going to play you a video that beautifully summarizes our findings. And yeah, give me a heads up, Keith, if this isn't playing for you guys. Cool. So that's a video that Jesse Kendall-Barr put together to sort of summarize the paper. But I think the main takeaway there is that, behavioral strategies are really important for elephant seals. And there's sort of mixed evidence, frankly, that clearly foraging success is really important for reproduction, less so for survival. And that, survival is probably at least in part driven by predation and probably in part due to starvation. So the next piece that I'm going to talk about is the fitness consequences of ocean conditions and trying to further disentangle those pieces. Starting with thinking about juveniles. Juveniles tend to be one of the more important demographic groups to think about in marine mammals because they have this really steep survivorship curve where the juvenile mortality plays a really large role in driving population persistence. So we wanted to ask how juveniles develop their diving abilities. We know in lots of large marine vertebrates that juveniles are less experienced, they're also smaller, and so in general, they're at this huge, disadvantage, whether it be physiological from having smaller body sizes and therefore lower oxygen stores, or whether it's from their, huge energy requirements because their mass specific metabolic rates are really high, or whether it's just the inexperience from not having done this before. And so we put instruments on a small number of weaned elephant seal pups. This was a postdoc that Joffrey Jomo did with me after Keith finished. And here's an example of what the tracking data look like. So here's a seal's first migration. But the piece that you can't see from the map is the diving development that's actually happening during this foraging trip. So we found that during the first trip to sea, the juvenile elephant seals started by holding their breath for, about 10-ish minutes. And then by the end of the foraging trip, they had almost tripled their breath holding abilities, which allowed them to dive a lot deeper and to basically have more foraging success, to gain more body condition, which is really important because we think that a lot of the mortality that's happening with the juveniles is probably due to starvation. One of the really interesting things that we've always sort of been puzzled about is that we tend to see these sort of population pulses in the elephant seals where there will be a few really good years and a few really bad years, and those tend to hit the juveniles harder. So we'll see years where, only, let's say 30% of the weaned pups survive their first foraging trip, and some years where it's 70%. So there's a huge amount of variation in how successful the cohorts are. And we wanted to ask whether ocean conditions might be driving those pulses. So I teach an upper division fields course. It's called Field Methods in Large Marine Vertebrates with these amazing students. And we allow them every year to choose a project that they want to pursue as sort of a course-based undergraduate research experience. And the teaching team, myself and two others, Allison Payne and Patrick Robinson, sort of guide them in the direction of a project that might be interesting. So 2 years ago, they wanted to look at the fluctuations in cohort success. They wanted to look, for example, at like the proportion of seals in a cohort that lived long enough to produce pups of their own or that recruited. And what they found is there was this huge amount of variation in the proportion of individual pups that recruited, ranging from 30% at the absolute peak to like 5% at the absolute lowest. So a 6-fold difference in the proportion of a cohort that lives long enough to produce pups of its own. And they brilliantly thought maybe oceanographic conditions, like a decadal cycling of some sort of index, might be driving these patterns via changes in food availability. And so they did their homework. They figured out that there's the North Pacific Gyre Oscillation Index, which is basically an index that's derived from, remote satellite-based measurements of different physical oceanographic parameters. And those are used to create this annual index that we can then associate with the elephant seal demography. And what they found was that when NPGO was quite positive, the seals did really well. When NPGO was really negative, the seal cohorts did not do as well. And so as a class, we basically traced the oceanographic conditions from surface-based physical oceanography all the way through the demography of the seals. So for example, we showed that different oceanographic indices, I showed you NPGO, but we also looked at PDO and SOI, so the Pacific Decadal Oscillation and the Southern Oscillation Index. These correlate really strongly with how much foraging success the pups' moms have during the preceding foraging trip, the foraging trip in which the moms are pregnant with those pups. Just a quick note that PDO is backwards. So it's a negative correlation because that's how that works. Very confusing. We then found that the moms that gained more mass produced larger weanlings. So there's this potential mechanism by which good physical oceanography cascades up to, biological productivity of some form, whether that's the fish or the squid that the seals are eating. The moms are doing better, and then they're producing higher quality pups. This is a little bit of a better sort of visualization of that. So here we have the relationships in a sort of path analysis between the demographic metrics. So between things like weaning mass and first-year survival and recruitment success are all very positively correlated. In the oceanography, so we see that NPGO with multiple lags, and also SOI and PDO are correlated with all of the demographic metrics of the seals. And I'm not going to go into it today, but in the paper that is in review right now, we basically use the association between these index values, which actually go all the way back to 1950, and the seal demography, those correlations, to project back the relative fish abundance in the mesopelagic in the North Pacific Ocean all the way back to 1950. So it's really difficult to measure relative prey abundance across years out in the open ocean. There's just not a whole lot of sampling that happens at that broad basin scale. And we're able to use the seals as sort of ecosystem sentinels to be able to sort of hindcast and even forecast the prey abundance in the open ocean, which I think is pretty exciting. In addition to, being able to use the seals as ecosystem sentinels, we're also interested in figuring out what is going on with their actual population. So I'm not going to spend too much time on these slides. They're extremely complicated, but I just want to show broad scale that using population matrix models, we can actually project the seal population, including the age structure, through time based on different hypothetical scenarios. So in this one on the left side, we said, okay, what is the difference between the best elephant seal cohort, which had 61% first-year survival, and the worst cohort, which had 18% first-year survival. So we calculated the age structure based on that, subtracted the two, and what you're seeing in this plot is the difference between the best and worst year. So for example, that big yellow dot is saying that in the age 1 seals, there are 26,000 extra individual seals, and the sort of diagonal line is then basically dying off throughout the years of the 20-year simulation, showing that annual mortality is reducing that cohort size, but that cohort is always a lot bigger in the best oceanographic year than the worst one. And then all the dots below are the pups produced by that cohort, them growing throughout their age structure and producing pups of their own. So we can do these sort of demographic simulations to show what's actually happening to the seal populations in good years and bad years, and how that might change in the future. And then we can take that age structure and actually calculate fish consumption. So we can calculate how much extra fish are consumed by the cohort, the good cohort itself, the offspring of the good cohort, and both of those two together. And we find that, in a good oceanographic year, the seals populations are so much bigger. They're consuming a lot more fish, 15 million more kilograms of fish than the weak cohort. And so this kind of explains not only why we're seeing population pulses in the elephant seals themselves, but it also explains why we might be seeing things like different rates of top-down control in, let's say, like fishery stocks that might happen in those good oceanographic years. Okay, so I started with this slide to think about the ecosystem consequences of living animals. I said that animals do a lot of different behaviors. They have a lot of different ecosystem consequences. And hopefully I've showed you that those behaviors translate to really important demographic consequences as well. So I showed you that life history timing has really important fitness consequences. We talked a little bit about the molt and how that's highly asynchronous across seals, but that they manage to become synchronous by the time the breeding season rolls around, both through phenological compensation, but also through really strategic turnaround strategies in the middle of their oceanic migrations. I also showed you that behavioral strategies can have really important fitness consequences. So for example, seals that dive deeper and forage more offshore and eat more fish gain a lot more mass, and they have higher lifetime reproduction and survival. And that they can avoid predation by their main predators, white sharks and killer whales, by strategically using three-dimensional space and time. And I also showed you that oceanographic conditions have huge amounts of fitness consequences, especially for juvenile development, but particularly across different generations, ranging from moms to their pups. I think one of the reasons I'm really excited about all of these sort of fitness components is that we can begin to understand the relative importance of them. What is the relative importance of ocean conditions or behavioral choices or physiological constraints of life history timing to reproductive success, and particularly at a population level, how do those factors control which animals are skip breeders, which are reproductive animals, and which don't survive to do either of those two things? And what are the transitions between those two? Likewise, how do they control what happens to reproductive animals? So all of those transition probabilities are really important for understanding population persistence. And I think understanding the mechanisms gives us a window into what we think might happen in the future. We can think about, trade-offs and what the environmental conditions might do to those trade-offs. So for example, in a good year, maybe there's limited trade-offs between things like current and future reproduction, or current reproduction and future survival, because resources are abundant, they're not limiting. And so the seals can really have it all. They can reproduce and they can survive and they can reproduce the following year. But you can imagine in a really bad year that there might be a really strong trade-off. And so animals might have to skip or might not make it to future reproductive attempts. So I think one of the major sort of directions of our future research is going to be incorporating all of these metrics together to see how environmental conditions mediate life history trade-offs at a sort of lifetime scale. One of the things I've been thinking a lot about is how we can better measure fitness consequences of variation. I think, today, hopefully I've made the case for long-term research programs and their importance. I think overall funding for them is declining and they're becoming more rare, which is a problem in terms of collecting data in sort of a natural experiment type way, including across lots of environmental regimes. So I think more long-term research, or at least pulling the pieces of long-term research that we have together, will be really promising for that. I think we need to measure on a lot more demographic groups. In marine mammals in particular, there are very few studies on juveniles. I guess adults are really overrepresented in the literature, especially reproductive females, because they tend to be really important to the population and because they tend to survive more. And so the cost of instrumentation is lower. But I think instrumenting males and juveniles is going to be really important to sort of flesh out that story. I think we also need to study in more geographic areas. We have a huge bias in biologging science, where a lot of the effort is in Europe and the US, and we need to really sort of diversify that. And I think also there's a lot of data synthesis work to be done. And I think open science is playing a role and will continue to play a role in how well we can combine our brains to better measure the fitness consequences of variation. I do want to put in a little plug that we, on my lab website, have these tracking data freely available. So if you're interested in checking out where our seals are, we actually have exactly 0 instrumented right now because our deployment season starts tomorrow, which is why I'm sitting in my office in Santa Cruz and not with you guys having a beer after this, unfortunately. But starting tomorrow, every day, one more seal will get instrumented all the way through June 5th. And so you'll see a bunch of instrumented seals and their live tracking data on that website. And it's pretty fun to watch their migrations unfold. There's also a Facebook page that gives research updates and stuff like that. So if you want to follow along, we tend to post publications and things like that on there. So with that, I just want to thank my awesome team. This is definitely not a solo effort. It's been a lot of work of a lot of people. Keith, I'm sorry I didn't take this picture. We probably have a screenshot of us over Zoom at some point, but yeah, I feel really grateful to be able to work with these wonderful people. And I think the last thing I want to say is just that, I don't have time to talk about it today, but the group does a lot more work on sort of diversity, equity, and inclusion in science. And I just want to plug sort of 4 of the publications that we've come out with in the last few years for folks who are either designing or leading field courses or leading interventions and sort of EEB departments. We have a couple publications ranging from how field courses can be really powerful vehicles for inclusion and equity, to how to make for safer field work, to how to sort of close the diversity deficit in oceanography. So I definitely recommend that you check out those publications. The bottom left one particularly was written by a few grad students of mine that I'm super proud of. It has some really clear, actionable recommendations for people across the faculty and staff and sort of administrator and student spectrum. So definitely check that out if you get the chance. And with that, I will just thank all the incredible people that I get to work with, including the field assistants and the funding agencies. And I will take any questions. Thanks for your time.
I'm not sure you can hear, but they are clapping for you. And so we'll pass that along. So I want to open it up to questions, and I want to start with Roxanne online. Go ahead, Roxanne.
Very cool stuff. Do you think that ocean impacts, PDO, NPGO, ENSO, are directly affecting prey availability or also impacting predators by reducing alternative prey?
That is the $1,000,000 question. Whoever asked that, I would love to work with you if you're thinking about those sorts of questions. So the short answer is we don't know. Because there's a really clear tie to the mass that seals are gaining during that foraging trip, we think that it probably has to do with the prey base. There's not a lot of trophic levels that happen between physical oceanography and seals. They tend to eat sort of myctophid fish that have a pretty simple, I think, trophic change under them. So we think that ocean conditions are changing the prey base and that seals are less successful because of that. We barely know anything about the predators that are eating the seals, especially offshore. We'll see seals come back with some shark bites that they clearly got really recently, like right off the coast. We saw last year a seal come back with a killer whale scrape mark on its back that somehow survived, but we really know barely anything about, predator-prey interactions in the open ocean, especially at depth. I think it's very reasonable to expect that oceanography would change predator behavior, just like it would change the abundance and distribution of the prey the seals are eating. But without any data on that, it's really hard to tell. One of the things my incoming postdoc is going to be working on is trying to disentangle the contributions of environmental variation to the demography of different age and sex classes. So like if certain oceanographic indices have more or less of a survival impact on, juveniles or on juvenile males or on juvenile females than adults, then we get a better sense for what the potential ecological mechanisms might be. But we have a lot of work to do in that area. It's a really good question.
All right, questions in the room. Okay, hang on, everyone.
Thank you so much for speaking with us. That was really, really interesting. I just had a question about the mechanism of skipping breeding. If that's
Yeah.
something like the female seals in bad condition just won't go to the colony at all, or if they breed and then if foraging conditions get bad, they terminate the pregnancy. Can you tell us more about that?
Yeah, that's a super great question, and again, one we're still grappling with. I think, historically we've wondered if they're not getting pregnant at all, or if they're pregnant and they lose the fetus. Some work that I was part of when I was a PhD student on Weddell seals showed that almost all the seals are getting pregnant and some are losing the pregnancy. So I think that's probably the case in elephant seals. We do know of some cases based on hormone analysis of seals that did not get pregnant, and we know of a lot of cases of seals that got pregnant and lost the fetus. So the sort of proportion of those instances is still a little bit unclear. You asked about behavior relative to that skip breeding, and most of the time that the seals skip, they do still haul out. We don't know why. We think, there's a lot of predation risk. There's no sharks or killer whales on the beach, so it probably makes sense for them to come back for the breeding season to at least get bred for the following year and also to avoid predation. But their life history timing tends to be really different, which is super cool and not something I've seen in a lot of other species. So the skip breeders will shorten that long foraging trip. Instead of 220 days, it'll be like 150 to 180. They come back in October, November instead of January, and then they stay for an unknown amount of time and then end up leaving and somehow getting pregnant. So it seems like their life history timing is off, but their behavior is relatively similar. I will say that this year we had a very weird thing happen, which is that we instrumented, like we always do, a bunch of adult females after the molt in June, and they're supposed to come back 220 days later during the breeding season. And one of the seals just didn't haul out ever. She hung out by Canada and then she kind of swam south. And we think we got 1 hour of satellite hits on land at Año Nuevo in March, and then she took off again. And I don't know if she bred or what she did, but literally she was on the beach for an hour, and she ended up hauling out like 5 days ago. And we ended up recovering her instruments, but she was at sea for almost an entire year and clearly skipped breeding. So I'm not sure if she lost her pup or what she was doing, but it's pretty rare for them to skip an entire haul out or to just haul out for an hour instead of an entire month. So that was a pretty weird case.
All right, looks like we had another question online. Go ahead, Roxanne.
Awesome talk, Roxanne. Have you noticed any morphological changes other than body conditions in low cohorts, such as smaller sized individuals? Also, have you noticed any bias in the sex ratio of pups born in poor quality years?
Yeah, it's a fantastic question. Um, Stephanie Adamczak she just finished her PhD. She was Dan Costa's grad student. I was on her committee, and she did a chapter that's not published, but it's in her dissertation about body size in elephant seals as a function of age and year. And she didn't see any major changes in body size across cohorts. You've probably heard of like the shrinking bird phenomenon that's happening. And she was sort of interested in whether that might be happening too, but didn't find any really strong supporting evidence. In terms of sex ratios, we also don't see a huge change in the pups that are born. Like I said, we don't know a lot about how the different sexes are differentially impacted by environmental conditions. So it may be that the age structure of like the males and females is offset during bad years, but we haven't looked at that yet. Maybe my incoming postdoc will be interested in doing that. The one thing that we really do see with sex ratios is that older females tend to produce pups that are more likely to be male than female, especially at the end of life. So I think we are seeing some sort of like senescence, like sort of late reproduction changes, but they don't seem to be associated with environmental conditions. They seem to be associated with later in the mom's life.
Interesting. Okay, question in the room.
Thanks, Roxanne. Super interesting work. I was intrigued by the section you talked about the ocean conditions and how that impacted these different cohorts in different years. And you brought up using these seals as sort of sentinels. And I'm wondering if you can expand a little bit on whether you are using the ocean conditions to predict the fish that you mentioned, or the fish to predict the seal cohorts, or the seal cohorts to infer the fish, and sort of what's the most measurable piece there and how that impacts your hindcasting and forecasting you mentioned.
Yeah, that's an amazing question. I feel like the paper's in review right now, and I feel like you're the reviewer too that I'm scared of. We'll ask the really hard questions. It's a really important question. I think what we ended up doing was basically trying to explain the amount of variance in the seal foraging success that was explained by the physical oceanographic conditions. And that's kind of what those bottom plots are. Sorry, I didn't put the R-squared values on there, but in short, the surface level indexes from the satellite imagery explain something like 40 to 50 to 60% of the variance, but there's still quite a bit of variance in seal foraging success that's not explained by the surface metrics. And so we sort of use that to say, look, we can't measure the fish stocks. Like nobody's going out into the middle of the open ocean and fishing in the twilight zone all the way across this, like, I forget, 5 million cubic kilometers of ocean that the seals can sample in a given year. The best we have is these surface level, indices, and it seems like the seals are able to explain some variation in the prey abundance that the surface level oceanographic metrics can't. And so the idea is that we would use at the very least foraging success as a way to understand what the fish stocks are doing above and beyond what the satellite remote sensing can tell us. And the demography then just ends up being a little bit of an extra piece to say, hey, also, the foraging success is having a really critical impact on the demography of these top predators. And this is just the elephant seals, but imagine what this sort of thing is doing to the seabirds and to the, things like beaked whales that we really can't, robustly study the demography for. So kind of encouraging us to think more deeply about the sort of population pulses that might be happening in other large marine vertebrate species as a result of what's probably, major changes in the resources that are available to them. But it's not a perfect system. And so I think we're looking forward to digging a little bit more into using seals as actual like biological oceanographers and putting things like, microsonar tags on them that can actually like have ADCPs that go and look at prey abundance in the open ocean. We just haven't done that yet.
Okay, we're going to try to get through a couple more questions, so I'm going to rush us along a little bit. Roseanne, go ahead.
Great presentation. How loyal are they to their individual sites? Are the beach populations expanding, or are they more panmixic?
Yeah, it's a great question. In short, elephant seals underwent a crazy bottleneck where humans hunted them to near extinction. There were 20 left in Mexico. Now, just over 100 years later, there are 240,000 elephant seals, and they've sort of recolonized the original colonies from Mexico all the way up north. So, they're actually like breeding all the way up into Washington and even into like British Columbia. And so for the most part, they're very loyal to their individual sites. But I think in general, the species is colonizing new areas, like more and more north. And that tends to be the juvenile seals that sort of prospect in their first couple of years before they actually produce pups of their own. So it seems like the populations are expanding. But they do a little bit of kind of back and forth moving and breeding across the different colonies for sure.
Okay, we're going to take one more question in the room, and then we're going to wrap up today.
Great talk, and I'm sorry to make you talk again about I was gonna say skip spawning, which shows my cards. So coming from like the fish perspective, I guess skip spawning is quite common in longer lived fish.
Yeah.
And I was kind of thinking about that in terms of a seal, and I remember that for fish, they're not fertilized, right? So they can reabsorb. So I guess what I am just maybe asking for some clarification on is thinking about the timescales of like if they don't have a pup, is that because they didn't actually breed on land? Because otherwise they are pregnant, right? Okay.
Yeah.
And then how long the gestation period is. And so maybe like at what, I don't know if they have trimesters. That's the only other mammal I know. But but I guess, yeah, just kind of like thinking about maybe like the time course of that annual cycle and how that all lays in. Thanks.
Yeah, it's a super great question. And yeah, God bless the fish people, because man, I've learned a lot about skip spawning that has helped me understand mammals, since we know a lot less about intermittent breeding or intermittent reproduction in mammals. I think, it's a really interesting question. And the short answer to your question is like, this is the annual cycle on the left. They're pregnant during that entire long trip. So they breed. Let me see if I can get you guys my mouse. They breed at the end of the breeding season. They go into this sort of like extended embryonic diapause, and then they implant during the molting period. Then they're pregnant that entire 10-month foraging trip. So yes, they have trimesters, kind of. It's just 10 months instead of 9. But if they're not going to reproduce, it makes a lot of sense to not like bring the fetus all the way to term and have this huge energetic expense throughout that entire foraging migration. Like I said before, we don't really know when that process happens, when the skipping happens. But the mechanisms are a little bit more clear, and we're doing a couple pieces of research to sort of try to get at it. I think that the conventional school of thought of wisdom is that seals are capital breeders. So, everything that they need to finance to like energetically finance the reproduction period, they're getting during previous foraging trips. They come onto land and that's it. They just use their body stores to energetically finance reproduction. And so the sort of predominant school of thought for a really long time has been that probably if the seals are not gaining enough mass to sustain a pregnancy or to get through the lactation period, then they're not going to breed. So maybe body condition is the sort of predominant driver of skip breeding, where bad oceanography means bad foraging success, means bad reproduction. And we did see that in the paper that Keith and I did, right? That foraging success and reproductive success are tightly correlated. But there's still some variance in that that's not explained. And I have this sort of crazy idea that I don't think is so crazy, that life history timing also probably plays a really important role in determining when animals skip breeding, because we see animals fall behind in the annual cycle. And if they can't catch up, they literally like miss the opportunity to breed. So think about like an animal arriving really late for the molt might not be able to get through the diapause period, or an animal arriving really late to the breeding season might miss an opportunity to mate with males. And if you take each of the pieces of the annual cycle and add them all up, they're almost exactly 365 days, which means if something takes a little bit longer than it's supposed to, let's say if El Nino conditions are really bad and the seals are doing longer foraging trips and getting less foraging success, that they may arrive late for the breeding season and therefore have to skip because of their life history timing, not necessarily because of their body condition. So I personally think there's probably two things at play. I think body condition and life history timing are probably interacting to drive reproductive success, at least in these capital breeders. But we have a lot of work to do to put the data together to figure out whether that's actually true.
Great. Roxanne, as we wrap up, can you take us back to the slide that has your contact information on it?
You got it.
And then folks can follow up directly with you if they have additional questions. But I'm going to try one more time for you to hear the applause in the room.
I hear nothing at all, but I really appreciate the thought.
Thank you so very. Did you hear any of that? No. All right. Well, the room was definitely telling you thank you very much. Thank you so much for being here, and good luck with your sampling and pegging season as you move forward tomorrow. And for everybody in the room, thank you so much for being here. We'll see you next week. Thanks, everyone.
Thanks, guys.
Thanks, Roxanne.
Thank you.
Great. Awesome. That was so cool seeing all the other studies that you've done.
Yay! We miss you very much, but that ecology letters paper will forever be the foundation of most of our lab's research.
I need to do something and listen to you outside of that speaker range. Maybe that'll help.
Yeah.
Yeah.
I now see the back of your head.
Nope, that did not help. I see what you mean. Yep. All right. Let's see. Is that better? Can I hear you? Can I hear you? I don't know yet. A little bit. Yeah, we're gonna have to catch up in person next time in California.
Yeah,
For sure.
that would be great. Hopefully next
Oh.
Friday, but I won't hold my breath.
Oh, yeah, Roxanne.
Hi.
This is Lee Torres.
Hi.
Microphone. Yeah.
Yeah.
Oh.
I can see you.
Hi. I don't know where to look, but thanks for an awesome talk. Yeah, really.
I'm now Vanna White again.
Yes. Hi. Yeah. Yeah, right. Anyway, nice
Yeah.
to see you virtually. Come visit us in person.
Yeah, thanks for coming. Sorry I can't be with you in person, but one day I can't wait to hear more about what you're doing. It's always amazing.
Yeah. Cool.
Yep.
Awesome.
All right. Thanks so much, Roxanne.
Nice to see you guys. Bye.
Bye.